Ischemic stroke is a clinical condition characterized by hypoperfusion of brain tissue and results in neuronal loss. Numerous evidences suggest that the interaction between glial and neuronal cells exert a crucial effect after an ischemic event. In order to explore potential protective mechanisms, it is important to develop models that allow studying neuron-glia interactions in an ischemic environment. Here, we present a simple approach to isolate astrocytes and neurons from rat embryonic cortex, and that, by using specific culture medium, allows the establishment of neuron or astrocyte-enriched cultures, or neuron-glia cultures with high yield and reproducibility. To study the crosstalk between astrocytes and neurons, we propose an approach based on a co-culture system in which neurons cultured in cover slips are maintained in contact with a monolayer of astrocytes plated in multi-well plates. The two cultures are maintained apart by small paraffin spheres. This approach allows the independent manipulation and application of specific treatment to each cell type, which represents an advantage in many studies. To simulate what occurs during an ischemic stroke, the cultures are subjected to an oxygen and glucose deprivation protocol. This protocol represents a useful tool to study the role of neuron-glia interactions in ischemic stroke.